All right, so that's what all these wires are. These are what allows me to record. I'm too cheap to buy anything that's actually good for recording, so I use what I have. All right. All right, so talking about the respiratory system. I want you to keep respiration different from ventilation. All right, respiration is the process of gases going places. Ventilation is simply air moving in and out of the lungs. So when we talk about the mechanics of breathing, we're specifically going to be talking about ventilation. When we talk about how gases are exchanged between the alveoli and the blood, and then the blood and the tissues, that's going to be different parts of respiration. So if it's going from our alveoli into the blood to get exchanged, that's external respiration because we're in contact with the external environment through our alveoli. Gases get transported. Yes. And then they go to the tissues, and then we have internal respiration. So think of that as going from the blood, gases going from the blood to the tissues. But also realize that gases also go from the tissues to the blood, right? So we'll talk about that as we move on. All right, then cellular respiration is at the cellular level. You need oxygen. Why do we need oxygen? To produce energy. And what's the byproduct of that we want to get rid of? CO2, right. A lot of people say ATP, and I'm like, that's energy. All right. And again, all of that happens, again, because you should have had this before, in the mitochondria. All right. Starting with the nose and nasal cavity. So your book for this is Guyton and Hall, Textbook of Medical Physiology in Clinical Key. That's where a lot of this information is pulled from. But a lot of the slides I pulled from my undergrad lectures in anatomy and physiology. So medical physiology books like to go according to this is how this works, this is how this works, this is how this works. Undergrad anatomy and physiology likes to go, all right, here's where the process starts, and here's the end product. I think that makes more sense. So I've tried to keep it that way. So that's why you see it organized a little bit differently than what you would see in your book. All right. Let me make sure I can do this. Red? Green? Yellow? Do you care? I don't give a shit. Okay, red. All right. I also like participation, so speak up. And if you don't like something, speak up. If I'm too loud or too soft, speak up, please. I also do not have the cleanest of vocabulary. So if that really bothers you, let me know, and I'll try to keep it on the down low, but dang. All right, starting with, anyway, back to the lecture. Starting with the nose and nasal cavity, all right, nose, we have the nostrils, that's where air goes in. We have hair in our nose, and that's to prevent large things from going in, not as large as a finger, but something like flies and dust and stuff like that. Primary layer of filtration. And then we go into the nasal cavity, and it's really weird. I recommend doing this in the anatomage table once you get to that section. Do a coronal section, and then peel back like a CT, and then you'll see something that looks like this, and you're like, ew, that's what we look like behind our face. It's really weird looking. So the reason why I put it here is we have these nasal conchae on either side. I don't like that. On either side, and then this septum right in the middle. So you have a right side and a left side. Okay, and that's where the air goes through. And so we have these. Anybody know why we have these conchae, also called turbinates, in our nasal cavity? Of course, they're lined with mucous membrane. But why do we have this kind of structure in there? Anybody know why?
It's like a flow, so when it's cold, it'll heat up the air in your sinuses.
Okay, one reason to heat up air or warm the air. And it does that through increasing surface area. So correct, both of you together, correct. All right, so you increase the surface area. That's why we have these. And of course, do you notice anything funny about that guy's septum, that person's septum? It's deviated to one side. Okay, so a lot of people have that problem. I don't know how this works. Nope, it doesn't get rid of them. This is actually my first time using this way. So if I'm like going, ooh, or how do I do this? That's why. All right, everything in a respiratory tract lined with the mucous membrane. Remember, a mucous membrane has that ciliated epithelial, pseudostratified ciliated epithelial cells that provide a layer of protection, and scattered through there are mucus cells, all right, or goblet cells, which produce mucus, all right? And as those goblet cells produce mucus, they go to the surface of the mucous membrane, and they capture stuff, right? One distinguishing feature of these pseudostratified cells is that they have cilia. Now that cilia takes the mucus, and this is cilia. This is literally how they beat, right? And they move the cilia move the mucus in one direction. So it moves everything, the mucus and everything trapped in it in one direction. If it's in your nasal cavity, it's going back, right? So you have that nasal drainage, and it's got stuff in it. You either spit it out or swallow it. Okay, because we want to keep things from entering our lower respiratory tract. Then we have sinuses. Sinuses, the only thing I'm going to mention about it, because this is more of an anatomy topic, you have 4 different ones. Maxillary sinuses, right here, frontal, sphenoid, kind of back here, and ethmoid, like right behind your nasal cavity. The thought about these is they reduce the weight of the skull and serve as a resonant chamber for the voice, but they also have openings, and I've highlighted a couple of them. It's just not working with me. How about that? Right here, into the nasal passages, into those conchae that we talked about. So sinuses do drain, whether you believe that or not when you have a sinus infection. All right, after we get past the nasal cavity, then we start entering the pharynx, and the pharynx has 3 spots. Just behind the nasal cavity, it's the nasopharynx. As you go down just behind the mouth, it's the oropharynx. And after that, you reach the laryngopharynx, where it divides into the larynx and the pharynx. There is no specific landmark dividing these two. It's whatever they're located behind, okay? Or whatever it's located behind. All right? The pharynx, like I said, splits off into the larynx and the esophagus. The esophagus is your passageway for food. Larynx is your passageway for air, sometimes water. I know everybody's done that before. Okay. Larynx is where your vocal cords are. All these cartilage structures I'm not going to hold you responsible for in physiology here. I just want to name them because they take part in the way that the vocal cords work. But the important part of this will be that you have all these cartilages make up the structure of the larynx and allow the vocal cords, which are right here, to do their job. And what is the job of a vocal cord? To vibrate. Okay? Vocal cords vibrate. Now here you see the opening here actually is called the glottis. And so something that's on top of the glottis that closes it when you're swallowing is called the epiglottis. Good. All right? You have false vocal cords on either side, and those just aid in creating a barrier so things don't go down into the larynx. All right. So the way the vocal cords work, these rigid tissues that are made up of, think of it something that's very similar to like like cartilage almost. Okay? Tough connective tissue, and they vibrate. When you pass air over them, they vibrate. When you put tension on them, they vibrate at a high frequency. So high tension. When you loosen the tension, they vibrate much slower with a longer wavelength. So just by tightening the muscles, you go, right? And that's how speech or sound is produced. All right, going from the larynx down, we reach the bronchial tree. All right? Once you reach into the trachea, you're technically in the lower respiratory tract. Okay? So when people talk about an upper respiratory infection, usually it's right here. If it's a lower respiratory tract infection, it's in your chest. Okay? We're going to start with the trachea. The trachea is the structure right here that goes from the larynx down to the primary bronchi. All right? There is a split right here, a very specific structure called the carina. All right? It's a cartilaginous structure, but it's also a very full of a lot of nerve endings in there. So if anything is aspirated and it hits the carina, you get irritated right away and you cough. All right? When you are suctioning a patient, if you ever have to do that, the way you know that you're in far enough is the patient will start going, and it's the scariest thing you've ever experienced in your life when you do it the first time. But you're hitting the carina. Has anybody ever done that before? You? Yeah, it's messed up, isn't it?
It's not great.
No. So, when I was in nursing school and we were practicing on a dummy, I was like, oh, yeah, yep, all right, pass my check off. Then they're like, oh, you're going to do it on a real patient now. I'm like, okay. And I did it, and he was like, and I was like, it's like, no, don't do that. That's the worst thing you can do. That's supposed to happen. So that's all driven by the carina right there. All right, so that's the bifurcation into the left and right primary bronchi. These cartilaginous rings around the trachea, their purpose, why they're cartilaginous rings, is to keep it open. But they are C-shaped rings. All right, there is only soft tissue behind it. And the reason why you have soft tissue behind your trachea is because that's where your esophagus sits. And when you are swallowing a bolus of food and it's going down that esophagus and expanding it, if there was a cartilage ring right here, you damage the cartilage ring. So you want to have soft tissue right there. So vacuum hose, because that's really what it's like, and behind it, the soft part to allow expansion. All right, our right and left primary bronchi go into our right and our left lungs. All right, that's the dividing point between our right and our left lungs. Now, what do you notice about the difference between the right and the left primary bronchi? Right one is straight down and much shorter. The longer, the left one kind of goes off at an angle and is a little bit longer. Okay? It's important to know that because most things that are aspirated will get aspirated into the right side because it's shorter and straighter. All right, if the primary bronchi divide into the left and right lungs, what do you think these secondary bronchi divide into? The lobes of the lung. All right, and how many lobes do you have? On the right side you have 3, and the left side you have 2. All right, because on the left side you also have your heart. I love the unity. All right, that's awesome. All right, and then you progressively get into smaller and smaller bronchi as you get closer and closer to the actual lung tissue, which is your alveoli. See, I like this a lot better than lecturing into a microphone with in front of a computer. Fucking hate that. All right, so going down to our alveoli. you notice that these smaller branches of alveoli tend to not have rings anymore, but they have cartilaginous plates to them. So that will allow it to still maintain a rigid, semi-rigid structure staying open, but allow it to be a little more elastic. All right? And why is that important? When air comes in, you need to be able to expand and contract. All right? Also, within this, you have smooth muscle, because during, let's say, a fight or flight situation, what do you want to be able to do? Get more air into your airways, and one of the ways to do that is to dilate these bronchioles. Okay. Okay. And then as you get further and further down to the basically what we call a terminal bronchiole, you don't have any cartilage. All you have is that smooth muscle and elastic tissue, and then you enter into the actual alveoli themselves. Okay, so that's the tracheobronchial tree down to the alveolar level. I'm so used to going back and hitting next on the keypad. That's why I keep walking over here. All right. So those smallest airways, the alveolar ducts or the terminal alveoli or the terminal bronchial trees, terminal bronchioles, they end in the alveoli. And what do I want to say about this? If you look at the alveoli, what do they remind you of? Grapes, like little clusters of grapes. All right, except these clusters of grapes are all connected to one another, and there are pores between them, so all the air that gets into one alveolus can be exchanged with all the other alveoli as well. Okay, why is that important? Because this is where gas exchange occurs. All right, and the walls of the alveoli are very thin. Does anybody know or remember how thin? One cell thick. All right, so you have one simple squamous epithelial cell between the outside air and the blood, or the blood vessel. All right, so if you want, we'll talk about this a little bit more in the next couple slides, but if you want to think about it, so if this is, back up, one cell, how many cells thick are a capillary? One cell. Okay, so if you have capillaries surrounding these alveoli and gas, say O2 coming here, it has to go through one lipid bilayer, 2, 3, 4. So it goes through 2 cells, but 4 actual lipid bilayers, right, to go through the cell. And that's the shortest distance you can have for gas diffusion. Stop doing that. All right. So I talked about this a little bit on a previous slide. Here's your alveoli, grape-like clusters. You can see the pores within them. And actually, if you look at a micrograph of alveoli, you can see these individual cells right here, and you can't really make out the pores real well, but they are there. All right, so that thin respiratory membrane, you have the capillaries surrounding each alveolus like that because you want to have a high ratio of gas exchange. And your alveoli not only have these simple squamous epithelial cells, which are also known as type 1 cells, but you also have these surfactant secreting cells. All right, so they are constantly secreting surfactant, and we'll talk more about that as we go along, but think of it as like a detergent that reduces surface tension. All right, you ever seen that experiment? They do it on all these kids shows where you put 2 glasses and you get them wet, you put them together and they're hard to take apart, but then if you put soap in it, they're easy to take apart. Same concept here. So alveoli, I had a demonstration for you guys, and I forgot to bring it. It's in my office. Alveoli, I think of them as like balloons, and if you get a balloon wet, the walls are going to be stuck together, right? Okay, so you want a way to separate those walls easily, and that's what the surfactant does. The third type of cell you have in alveoli are alveolar macrophages. And I want you to think of these as like little Roombas within the alveoli that keep it clean. So if anything actually does get past the nose hairs, the ciliated mucous epithelium, or anything else that's in your respiratory tract to help defend, these guys will take care of it. Okay? So these are your final stop to try to get rid of anything that enters the lower respiratory tract. Okay. A little bit more about the surfactant. This picture right here shows two alveoli. One is small, one is large. A little bit about the physics of this. If you have a small alveolus, when you get air in there, it's going to expand much more than a large alveolus would. Okay? So, without surfactant, when air goes in and you expand these two, there's going to be an unequal distribution of air going from the smaller one to the larger one simply because it's recoiling faster and pushing air not only out but into this other one. That's one of the reasons why it's bigger, in fact. If you have surfactant, that reduces the surface tension and doesn't allow that expansion to occur as much. Okay, so you have an equal distribution of airflow into both alveoli.
Can you speak up a little bit when facing the screen?
Yes.
Is there like a lower setting?
Yeah. It's a constant battle. Do we stay cool, or do we? And in the winter, it's do we stay warm or do we listen? That's actually better. That was noisy. No, I'm really hot. I'm hot. I'm going to be hot anyway, so it doesn't matter. Alveolar gas exchange, the pores. We talked about that. Alveolar macrophages, respiratory membrane. We talked about all this. This is an electron micrograph right here, and it's showing the pores between the alveoli. So although you can't see those pores in like a light microscope, once you get down to the electron microscope level, it allows you to see things a little bit better, and you see how well connected these alveoli are. Okay, so what's the function? Why do we have these air sacs filled with air and these capillaries surrounding them? The whole reason? Gas exchange, right? So, remind me, at this level, what kind of respiration is it? External. Sweet. Okay, so air's here. Air is a mixture of oxygen and CO2. It's a mixture of a lot of other things too, but those are the two we're going to be concerned about. In fact, what's our air mostly made out of? Nitrogen. I was thinking about this the other day. I'll go off tangents, so don't let me go too far. You know those gas, if somebody can tell me, I haven't looked it up, tell me the reason for this, that say, we can fill your your car up, your tires up with nitrogen. You ever seen that? The nitrogen, we'll fill it up with nitrogen. Like, our air's already like 78% nitrogen. How much more is that 20% going to give me? Like, does anybody know the reason for this?
Isn't it because like when your tires get warm or cold, there's less expansion or traction within their gas versus like in your gas where there's oxygen and carbon dioxide?
Don't know. I don't know.
Oh yeah, because when you have the weather, so like one of my tires will be a little bit lower tire pressure, and that's just because of the weather, and then how they inflate and fluctuate based on the weather outside, if it's cold or hot. So you're minimizing that expansion the same way
So 20% makes that big of a difference?
It's like their detergent, like you explained.
Okay. Thank you. All right. Where was I? So we have this mixture of oxygen and carbon dioxide. It's at a certain percentage. We don't care what that percentage is by itself. We care what those percentages are in relation to the percentages of those gases in the blood. Because what drives all processes in biology? Gradients. Gradients. Okay, yes, homeostasis. But these gradients allow for homeostasis to occur. Okay, so you have gradients. So in the alveoli, you have a relatively high, and in fact, through all of this, I don't know what you did in undergrad. I don't care what you did in undergrad. I don't expect you to know the numbers. It's the concept I care about. So I'm not going to say, you need to know that it's 104 blah, blah, blah of oxygen in the alveoli. No, it's higher than the blood. Okay? So higher oxygen, low CO2, but in the blood we have low oxygen and high CO2. So we have a gradient. You're going to go out or in, and you're going to go out. Right? Simple as that. I find that when you start putting numbers in there, unless it really serves to drive the point home, it kind of muddles the concept. So I don't like it. I hate physiology too. I'm not lying. I love anatomy, hate physiology. But I've been doing it for 20 years, so. So what are the lungs? Simply this hanging bunch of alveoli in our thoracic cavity, right? And if I would have thought about it, I would have brought the pig lungs in here too. But I have to steal them from the science lab, and I don't always know when somebody's going to be there. I'd like nobody to be there so I can walk in and walk out with them. You didn't hear that? All right. Surrounding those lungs are the visceral pleura. So you have this membrane surrounding the lungs called the visceral pleura. Visceral meaning on the organ, so lining the organ. On the other side of that, you have the parietal pleura. Parietal means wall, so that's literally attached to the thoracic cavity wall. And between those, you have the pleural cavity with serous fluid. All right, so that fluid. And what does that fluid allow those two membranes to do? Stick together. One, and slide past one another, reduce friction. All right. The fact that they stick together is a pressure that's applied. So if you try to pull them apart, it's difficult to pull apart. We call that pleural pressure. Okay? So what happens is if we start, so through that way, the thoracic wall and the lungs are connected to each other. If you start pulling the thoracic wall out, because of that surface tension, that pleural pressure, you're also going to pull the lungs with it because it's pulling on those two membranes. Okay, so that's kind of what pleural pressure is and what we mean by intrapleural pressure, so within all the pleura of the lungs. Does that make sense? More or less. All right, and then of course the lungs are divided into lobes. We talked about those. You guys seem to be well versed in that. You just had it in anatomy, so no surprise. All right, and there's a picture of the lungs. It's really funny. When I do the digestive system, and I think I'll do it, I do it for a graduate level too. At the end of digestive, I have the rectum and anus, and I make a joke about how there's a giant asshole on the screen. All right. Now let's get to so that was our introduction. Now let's get to the physiology of things. How does all this work together? Now that we've set up the stage. Inhalation or inspiration, we apply Boyle's law to this. All right, and Boyle's law for gases states that as you increase volume, you decrease pressure. All right, so P times V, pressure times volume, equals a constant. In order to keep this constant, when one goes up, the other has to go down. All right, that's a balance. All right, remember we said in our mouth, all the way down, or nose, all the way down into the alveolar sacs, that's all external, right? Because it's in contact with the external air. So at rest, when the lungs are at rest, the atmospheric pressure, which is about 760 millimeters of mercury, outside is the same as it is inside. So they're equal. When you have equal air pressure, is anything going to move? No. Okay, so nothing's going to move. So, but we know we breathe air in. And we know it's because of a difference in pressure. So how do we create that difference in pressure? Okay, that's the process of inhalation. All right, the primary muscle involved in breathing is the diaphragm. As a longtime science teacher, I get like spasms when I hear people say, my lungs are working really hard. Because your lungs don't do shit. It's your diaphragm that's doing it. So, diaphragm contracts. All right, and when a diaphragm contracts, it pulls downward. All right, and it compresses abdominal organs. The other primary muscle involved are the external intercostals, which are, can you see them here, or can you see them here? Remember from anatomy? Did you pay attention to that? All right. You can see both the externals and internals from the front. You can see them both. Can you see externals here or externals here? Here. Right. So, externals contract. They elevate the ribs. So not only do you have the diaphragm pulling down, but you have these external intercostals pulling the ribs up, so you're expanding your cavity. All right, tell me, because of the pleura, what's happening to the actual lung tissue? The lungs are going to follow, right? So by doing that, you're increasing pressure or volume? You're increasing volume. All right? If you increase that volume by Boyle's law, what has to happen to the pressure? It goes down. Atmospheric 760, your lungs go down to about 758. That's a big enough difference in pressure to suck the air in. Okay? There's the number. Is that going to be the same in everybody? No. And if I use all my respiratory muscles and expand farther, is that pressure difference going to be bigger? Yes. Okay. And the faster I do it, the greater velocity with which that happens as well. So this is, generally speaking, quiet breathing as you're sitting there being bored to death by a physiology lecture. All right. We also know that we can take a deep breath. This is going to come up later, but I'll mention it now. When we're normally breathing, like you guys sitting here, what's the volume of air that normally comes in and out called? You guys remember? Tidal volume. Good. Tidal volume. Anybody know what about, on average, the adult tidal volume is? Nope. About 500 milliliters. So about half a liter. You're off by an order of magnitude. So yeah, 500 milliliters. That's when we're using our diaphragm and our external intercostals. If we want to take in deeper breaths, which would actually be our inspiratory reserve volume, which on average is about 3,000 milliliters. Then we're going to use, we can contract the diaphragm even more. We can use our pectoralis minor muscle, which is underneath our pec major, so it's a little muscle going from here to here, transverse, so it's going to pull things up. And our sternocleidomastoid. Watch somebody take a deep breath. You can see my sternocleidomastoid is coming out, right? It's pulling my rib cage up, expanding my thoracic cavity even farther. So these are what we call accessory muscles that allow for deep inhalations. All right. We talked about the surface tension in the pleura, right? That allows our lungs to expand. So I don't think that, okay, let me ask you this though. All right, I have to explain something else first. We also have surface tension in our alveoli, right? Our alveoli want to collapse because it's wet, right? Anything that's wet wants to stick together. What do we use to prevent that? Surfactant. And the fact that our lungs are always filled with a little bit of air. Okay, so our lungs are always filled with a little bit of air, and we have that surfactant. Despite that, our lung tissue, the alveoli, are made out of elastic tissue, so they're going to want to collapse. That surface tension, there's still going to be some there, and the elasticity want them to collapse. So our lungs are always moving towards collapse. Outside of the air that's already in there, what's another thing that keeps them open at rest? Like if I'm not breathing. Which pressure? Interpleural, thanks. Great. The interpleural pressure. So think about this. Our lungs want to collapse, but that interpleural pressure is preventing them from collapsing as well because it's holding our lung tissue against the thoracic wall. Okay? So that's a pressure. When we take a breath in and our lungs expand, now our lung tissue is like pushed right up against our thoracic wall. What happens to the pressure in the pleura? Do you think? We're putting it right up against the wall. Like we're not pulling on it anymore. It's like right up against the wall. Pressure goes down. Does that make sense? So if I have my left hand pulling my right hand, right, inflating this little space, and I inflate my lungs and then I pull it all the way up against, there's less pressure that has to hold it there because this is actually pushing up against it instead of this just pulling. Does that make sense? So think about that. So interpleural pressure goes down. And your intraalveolar pressure is also down because volume went up. Okay, we're talking about two different physical processes that are going on. Okay, so you have the intrapleural pressure keeping our lungs open. You have the intraalveolar pressure trying to collapse our lungs. And both of those working against each other is what helps us inflate and deflate our lungs, that surface tension. If we actually have alveolar collapse, if we lose that surfactant or we get air in there that pushes our lungs away from our thoracic cavity wall, we have a lung collapse, which is called atelectasis. So if you hear that word come up, and you will in your clin med classes, atelectasis is a lung collapse. All right, so how do we exhale? Like we've been sitting this whole time going, all right, come on. Boyle's Law, again. So we increased volume to get air in. So what do you think we have to do? Decrease volume. Okay. Now, what caused us to increase the volume? Diaphragm contracting. So what's going to allow it to decrease? Relaxation of the diaphragm. So as soon as you relax the diaphragm goes back into position. Those abdominal organs that you push down recoil up, right? What happens what happens to your thoracic cavity? Volume goes down. So what do we have control over, volume or pressure? Volume. Pressure is a function of the volume that we control. Okay, we control the volume. Pressure comes afterwards. All right, so we relax those. And so volume decreases. As volume decreases, what happens to pressure? Pressure increases. So maybe you have like a 762, 760 out here, air is going to flow out. Okay? So differences in pressure are allowing the airflow by us controlling differences in volume. Okay? So, I don't want to hear any of you say lungs are working hard. You will anyway, because I do it too. Even though, those things you rail against, but you're like, all right. So we relax those muscles, but when we exhale, can we always exhale more? I don't recommend doing that. We can we can always exhale more. Thank you. I forget it's there. Part of teaching is being completely okay with making an ass of yourself. So, not only does relaxation allow you to exhale, but we can actively exhale more, as I just demonstrated, by contracting our internal intercostal muscles. So, external for inhale. It would be nice if they were reversed, right? External exhale, internal inhale. External for inhale, internal for exhale. Okay? So they pull ribs down and inward, pushing air out, and we can contract our abdominal wall muscles. When we contract those, what happens? What do you think happens? We talked about it during inspiration, but when I contract these muscles, I'm pushing all my abdominal contents up, pushing that diaphragm up, and forcing more air out. Okay, so this is kind of a summary of the pressure differences that are happening in our lungs and the volume. So this red right here represents our lung volumes. And of course, here is inspiration, here is expiration. I like inhalation and exhalation because inspiration to me means like I'm inspired to give a great lecture, whereas expiration means I feel dead afterwards. So inhalation, exhalation, volume goes up. That makes sense, right? In the lungs, lung volume goes up during inhalation. But what happens to alveolar pressure? It goes down. So this stuff has been measured, and this is how we know it. Alveolar pressure goes down. What happens to your intrapleural pressure? It also goes down, right? Because that lung tissue is now pushing up against it, so it doesn't have to pull as much. All right, so those pressures go down, and then in the process of exhaling, of course, air comes out, and those pressures go right back up to resting. Okay? So this is just a visual of all the stuff that we have been talking about. All right, now let's go into the pulmonary blood flow. So those are the mechanics of breathing, also known as ventilation. Good. Now we'll go into the blood flow. So we have to have blood flowing in and blood flowing back out. All right, so our lungs are theoretically divided into 3 zones. The upper zone is zone 1, about the top third of the lungs. Zone 2 is in the middle third. Zone 3 is the lower third. All right? In your upper airways, I'm listening to a podcast right now called Star Talk Radio. Anybody ever listen to this? Neil deGrasse Tyson? You know who he is, right? Physics guy. So, like, I'm a super nerd. This is what I do for fun. So I'm listening to this, and he's always talking about physics. If I were to go back to school and do things, I'd do chemistry or physics because they're fascinating. I just didn't have the confidence in myself back then, so I became a biology teacher. I'll tell you guys that story someday. My original path was medical school, actually. Zone 1, Zone 2, Zone 3. Oh, physics. That's right. That's where we were. All right. Simple laws of physics. Newtonian physics. Air is light, so it's going to go up. Air is light. It's going to go up. Fluid is going to go down because it's heavier than air. All right? So, in the upper airways, you're going to have really big alveoli because air, when it goes in, it's going to float up. Okay? And they're going to be more expanded because of they're under higher pressure because air flows up. All right? And because they're higher expanded, you're going to have less blood flow going in there. Okay? Partially because they're expanded and compressing vessels, also because blood's working against gravity, so not as much is going to go up there, right? Does that make sense? All right, so high airflow, low blood flow. In the middle, you got about equal amount of air as you do to blood flow. All right, so blood flow and airflow are going to be functionally equivalent, is what we'll say. They're going to be functionally equivalent to each other. And that's what you want. And then you can adjust the respiratory, the airway to allow more air in or less air in, and the blood, the capillaries, to allow more blood in or less blood in. So you can modulate airflow and blood flow at this level, mostly blood flow. And at the bottom, air doesn't want to go down, it wants to go up, right? So you're going to have less air going down, but more blood flow simply because it's easier for blood to get into the lower lobes, right? Because of gravity.
So you have the most pressure on the bottom. Like the intravascular.
In terms of blood flow? Yes.
Yeah.
Yep. You're going to have the greatest blood pressure at the bottom. Now, I mentioned at the beginning, Zone 1, Zone 2, and Zone 3 are theoretical zones of our lungs. Zone 1 in a healthy lung does not exist. Okay, that's going to be important to our discussion going forward. Zone 1 does not exist in any of your lungs right now, unless you have a respiratory disorder that I'm not aware of. Okay, so that means most of our lung is Zone 2 and Zone 3. It's Zone 2 from about the apices of our lungs to just above the heart, and Zone 3 all the way the rest down. All right? So what that tells you is we get adequate airflow and adequate blood flow. You could also make the argument that we get an overabundance of blood flow. Is that good or bad?
It's bad, like to a certain extent.
It can be bad, but mostly good. In healthy lungs, it's good. Okay? All right.
Professor Ways, it is 2 p.m.
Can I finish this one first and then
Yeah.
break? Okay, sweet. Thank you. What slide number are we on? 27? And we're almost halfway. 58?
Yeah.
Okay. All right. From our heart we haven't covered this yet, but I'm assuming you guys are all reasonably intelligent people because you got into PA school. And you've had some physiology before. From the right side of the heart, we pump blood where? To the lungs. And those are called pulmonary arteries. Good. All right. When blood comes back from the lungs into the heart, left side of the heart, it comes through the pulmonary veins. Now, left side of the heart pumps out to the rest of the body, right? That, we always say that oxygen levels in arteries is usually high, right? But in the pulmonary arteries, it's just the opposite, right? Because it's coming back from the body, going to the lungs, so it's very low in oxygen. All right, and then coming back from the lungs, the veins are high in oxygen, which is the opposite of what, the rest of the body does. All right. You would think that along those ways and through the trachea that some of our tissues in the airways would get oxygenated, right, and get rid of carbon dioxide, but they don't. Okay? So our tracheobronchial tree, being a living organ, has its own dedicated blood supply. It doesn't come from the pulmonary arteries or pulmonary veins, right? It comes from separate arteries, which are called bronchial arteries. You see, one of them comes from the actual aorta, and then other ones branch off from the pulmonary veins. But those big vessels don't give the actual trachea and bronchioles their blood supply. They have a separate blood supply. So that's what this slide is meant to tell you. Okay? Living tissue. Even like, what's this right here? The aorta. Big ass blood vessel. The blood that's inside of it does not feed the aorta itself. The aorta has its own little blood supply within it to feed it. Okay? So this concept is going to exist through all of anatomy and physiology, is where like things that you think would have blood flow from a certain place have it actually coming from their own dedicated blood supply. All right? So let's take a break and 5 minutes. All right, I will tell you guys something. Every clock in this building has a different time on it. So you cannot judge time in class by what's going on the wall. All right. Let's continue. Dead space. Probably how you feel right now, right? All right. Dead space is a normal phenomenon in our lungs. It's exacerbated in unhealthy lungs. But the definition of it is the volume of air in the airways that doesn't reach alveoli. There's different kinds of dead space, it turns out. That's called anatomical dead space because they're anatomically stuck somewhere, right? And that's in the airways. If it's in the airways and not in the alveoli, there's no way for that gas to get exchanged. So that's what we call anatomic dead space. That's about 150 milliliters. Do you remember what tidal volume is? 500 milliliters. All right, so if every breath you take in is 500 milliliters and 150 of that never takes part in gas exchange, then only 350 milliliters of air is available for exchange. Does that make sense? Is that wasted? Or does that serve a purpose? Keeps it from collapsing, right? So the primary purpose that would serve is keep things from collapsing. All right. There's also a volume of air in the alveoli that does not get exchanged because of poor blood flow. We call that physiological dead space. All right? If you can't exchange because of poor blood flow, you have physiological dead space. So that an example of that would be zone 1, right? Low blood flow, gas exchange can occur. In normal lungs, physiological dead space is 0. Okay? So that's what it means when it says anatomic dead space equals physiological dead space. In a normal lung, physiological dead space is 0 because you should be having adequate blood flow everywhere in your lungs. Okay? So it's only in pathological conditions where that physiological dead space will become important. Okay? So anatomic dead space plus alveolar dead space equals physiological dead space. Did I say that wrong? I did, didn't I? I jumped ahead of myself. Air in alveoli that does not get exchanged because of poor blood flow is alveolar dead space, not physiological dead space. If I do some shit like that, call me out. Like if you're like, what the hell is he saying? Like, good thing I caught it. All right, so alveolar dead space. So anatomic dead space plus alveolar dead space equals physiological dead space. All right? Since alveolar dead space should be 0 in a normal lung, that means anatomic dead space would equal physiological dead space. That's a better argument. As a teacher, you also learn that it's okay to be wrong, as long as you explain yourself. All right, alveolar ventilation, we have minute ventilation. So that's the amount of air moved into the respiratory passages per minute. We get that by multiplying our tidal volume times our breathing rate. So if our breathing rate is 10 per minute, what's our minute ventilation? 5,000. Okay. What's normal breathing rate? 12 to 20. 12 to 20. Actually, and you guys, anybody here like do patient care stuff now? Okay, or has done it recently? Okay. Does anybody actually count breaths ever? No, never.
Yeah,
Right. You don't start counting unless you note it. Like, I work at a drug rehab clinic, and a lot of them are opiates. What do opiates do to your respiratory system? They suppress it. So if you see somebody sitting there going, then you want to start counting. Or if they're going, then something might be up. But generally, when I get my, so we have our mental health techs. I work twice a month on the weekends. We have our mental health techs. They get all the vitals, and they give the nurses the vital sheets. And I looked on my vital sheet, and it says 16 for everyone. I'm like, you're not fucking counting anything. So, because it's difficult to count, it will take forever if you actually sit there and count it and try to get their pulse and everything. So, best guess. But if you see something off, like Gabe said, then you really want to start counting. Okay, there's what they teach you in school, and then there's reality. Okay? Alveolar ventilation rate. So then we'd be talking about what is actually getting exchanged at the level of the alveoli. That would be your tidal volume minus your physiological dead space, which in a normal lung is equal to what? Anatomic dead space. Right? Which is not 0. Okay? This is where that mix-up occurred. Times breathing rate. Okay? So that would be the 150 then. times, or 350 times breathing rate. So 350 times 10, that would be 3,500. So actual alveolar ventilation rate compared to minute ventilation is a 1,500 milliliter difference, right? That's not getting exchanged. All right. Now we want to get into the concept of ventilation versus perfusion. All right. There are letters associated with this. Ventilation is a V. Perfusion is a Q. And I was thinking about this. So like, why can't it be a P? Can anybody tell me why it couldn't be a P? Because pressure is P. And pressure came before perfusion, scientifically speaking. All right. We already established that our respiratory membrane is thin, literally 4 lipid bilayers. All right. And gases diffuse from regions of higher partial pressure to lower partial pressure. All right. So it's not just ventilation that pressure is occurring. It's also in the respiration processes that pressure differences drive movement of gases. All right. So at the level of the lungs, because of this, relatively high, well, we'll use the numbers. I don't care that you know them. I'm just using them to serve a point. In the lungs, oxygen is 104. In the blood flowing into the lungs, oxygen is 40. Which is lower? 40. So oxygen's going to go that way. All right. In the lungs, CO2 is 40, but in the capillaries, it's 45, which is lower. 40. Carbon dioxide is going to go the other way. All right? You're simply moving from higher numbers to lower numbers. I mean, it doesn't get simpler than that. You don't have to know the numbers, so don't ask. Somebody will. That's fine. I'll still answer you and not make you feel stupid. I won't. I'm really, you guys will find out I really go out of my way to not make you feel stupid. So if I ever accidentally do that, tell me about it because I didn't mean to do it. Unless you piss me off. Don't piss me off. All right. So why we mention this, ventilation-perfusion ratio is an important concept when you're talking about respiratory physiology. All right, so it's your V over Q, ventilation-perfusion ratio. Okay? Normal VQ, averaged across all zones of the lungs, is about 0.8, which means it's close to 1. So what does that mean? If it's close to 1, then ventilation is equal to perfusion. Okay? All right. A little bit of math here. If it's 0.8, what's the higher number? That doesn't equal 0.8. I get that. It's just for demonstration purposes. Right. So the lower number would have to be higher, or the Q would have to be higher, right, in order to get a 0.8. So what that means is we have a certain amount of ventilation and more than adequate blood flow, right? Okay, looking at the zones. All right, before we go through, I'm doing this like you guys can see my hand. Before we go through this, we're going to discuss zone 1, 2, and 3. Does zone 1 exist in a healthy lung? No. Okay? We're still going to talk about it, though, as a concept. So everything we're talking about here are conceptual things to think about when you're referring to how ventilation and perfusion occur in the lungs. It doesn't mean that it's happening. Okay? So you see, these are what we call proof of concept. All right, zone 1. What do we know? Lots of ventilation, right? Low perfusion. What's that going to give us? That's going to give us a high VQ ratio. High V, low Q. High ventilation, low perfusion. Okay? Excuse me. What's really happening here is you have very low ventilation because these air sacs, they're large, so not much air is going in and out of them. But you have much lower perfusion. That's what's really happening in that zone. So it's not that you have a high ventilation and a low perfusion. It's that you have a low ventilation but an abysmal perfusion. Okay? When we have this situation where we have low blood flow and high ventilation for all intents and purposes, we call that physiological dead space. Right? Look at this coming together. Right? So zone 1 would be physiological dead space. Lots of air, very little blood going in there. Okay? Zone 2 would be our 1, right? Ventilation and perfusion are equal to each other. Zone 3, we would have, remember, low ventilation but high perfusion. Okay, but what is really happening here is you have high ventilation, really high perfusion. The reason? Small alveoli that can expand. They will allow more air to go in and out, right? So high ventilation, really high perfusion because of gravity, all right? So here, potentially, you could have so much blood flow going through that gas doesn't get exchanged effectively. We call that shunting. All right, so in zone 3, shunting can occur. Shunting is passing through without exchanging gases because you have too much blood. I was going to ask you guys a question. What was it? I don't remember. Fuck it. All right. So, does that make sense? All right. Now, most of our body sits, or lungs, rather, sit kind of there, right? It's only in pathological conditions where we're going to have these things occur. Okay, and we'll talk about some of those pathological conditions towards the end of this when we talk about lung compliance. All right, if that explanation didn't work for you, watch this video, okay? It's either on here or it's one of those videos that I put in Canvas for you to watch. All right, this guy gives a good explanation of ventilation perfusion ratios and what they mean. Oops. I'm really picky about the videos I show, so I wouldn't show it if it wasn't a really good video. So, all right. So that's what allows us to get, control how much gas is exchanged within the lungs. Is that ventilation perfusion? Oh, that's what it was. Every once in a while. Neurons fire in my brain every once in a while. Okay. If ventilation is equal to perfusion here, we call that a ventilation perfusion match. If ventilation does not equal perfusion, and this is what you'll hear clinically, then you have a ventilation perfusion mismatch. Okay, so that's the term you will hear, is a ventilation perfusion mismatch. And when you hear that, this concept is going to come into play. All right? All right, since I'm on the subject, can a disease cause a problem with ventilation at the level of the alveoli? Yes. Any? Cystic fibrosis. Another one? Emphysema. Good. Okay. Can a disease produce a problem with perfusion? Diabetes would be an example of that. Okay. So, depending on which disease you have, you could be affecting ventilation or perfusion, producing a ventilation-perfusion mismatch, and then you have to correct for the one that's wrong. Okay? So that's where this concept would go. I'm glad I remembered that. All right. Oxygen transport. All right, so we talked about, oxygen came from the alveoli, got into the blood, you got rid of carbon dioxide, okay? So when oxygen is in the blood, almost all of it, 98%, is attached to hemoglobin. Okay, you guys all have heard of hemoglobin before, right? How many oxygens can attach per hemoglobin molecule? 4. Okay, good. So when up to 4 are attached, that means that hemoglobin molecule is saturated. Okay? And usually, when we drop off oxygen to the tissues, how many oxygens come off of the hemoglobin? Typically, under normal conditions. Usually just 1. So then you still have 3 left, and you go back and you just pick up that 1 extra. So under what conditions might you drop off more oxygens from hemoglobin? Exercise. Fight or flight situations. Disease conditions, right? Would that don't allow you to get adequate perfusion of tissues, that might happen. All right. So when oxygen levels are high, and in the lungs, are they always high? In healthy lungs, are they always high? Yes. The answer is yes, for those of you looking for it. When oxygen levels are high, oxygen binds tightly to hemoglobin. We say that has a high affinity. Okay? When oxygen levels are low, and that happens in the lungs, when oxygen levels are low, like in the tissues, affinity drops, oxygen goes off. Okay? So, if 98% of the oxygen is carried in hemoglobin, where's the other 2?
Dissolved in the blood.
Dissolved. Yep. Dissolved in the blood. All right. If we want to increase the release of oxygen from hemoglobin, we either increase carbon dioxide, which is what would happen in our muscles during exercise, or in our muscles during a respiratory condition where we can't get enough oxygen. Increased acidity, which is what's going to happen during exercise too, like the lactic acid buildup. Or an increased temperature, which if you're exercising, what's going to happen to your body temperature? Increase. So all of that's going to increase the amount of oxygen that gets off of hemoglobin, that gets released. So then you'll start releasing more. That means you have to go pick up more. All right. So we talked about this. This is in the lungs. This is in peripheral tissue. All right. Oxygen goes here, attaches to, notice there are 1, 2, 3 oxygens on that hemoglobin. Now there's 4. Quick question. If oxygen is always 104 here, and it's 40 here, so you have oxygen going in, when does this diffusion stop? Pardon?
When it reaches 104 or 140.
When they're equal. When the pressure here is equal to here, so it would be about 140. Right. Or 104, sorry. Mixing the numbers up. So, I forgot to mention that earlier. Okay, so that's when, here's another question. and has nothing to do with this class, but it's an important concept to remember. As blood's moving, you always have low oxygen here, right? You always have low oxygen coming in. If blood were to stop and diffusion comes to an equilibrium, what would happen?
Stop circulating oxygen.
And then you die. So I always teach my biology students, because they have this, or my A&P students, undergrad, equilibrium equals death. All right? Equilibrium is not the same thing as homeostasis. Equilibrium equals death. Okay, you don't want things to be equilibrium, which is why you make gradients all the time. And the flow of blood creates a constant gradient. Okay? All right, once you get to the tissues, you have high oxygen in the blood, low oxygen in the tissues. Oxygen's going to pop off and go into the tissues. Okay? That makes sense. And like we said, you only get rid of one. Okay? So why do we have 4 attached if we're only getting rid of 1? Pardon? For when we need it. We call that reserve capacity. Okay? Our body has a huge reserve capacity of everything. This is just one example. All right.
Like a bit off topic, but like for example, if someone's like hypoxic,
Mhm.
and their oxygen is like, does that mean they have trouble getting the oxygen off the hemoglobin? Like, or is that kind of like that's like not really like correlated with each other?
It could be. So hypoxic mean. Here's a terminology lesson for you. Hypoxemia is low oxygen in the blood. Hypoxia is low oxygen in the tissues. There's a difference between those two, even though everybody uses them interchangeably. Okay? So hypoxemia, or hypoxia specifically, could mean that you don't have enough blood circulating. Maybe you're not getting it off the oxygen. There's something wrong with hemoglobin. But usually it's lack of blood flow.
What did you say perfusion stops again? Perfusion stops when it's at equilibrium, the pressure.
Oh, so diffusion.
Oh.
Diffusion stops when blood flow stops. Okay? Diffusion stops when there's an equilibrium. All right. I know everybody loves oxygen hemoglobin dissociation curves. I hate them. Basically, what this shows you is a curve of how oxygen comes off of hemoglobin in a physiological context. All I want you to get from this is that when you have increasing levels of CO2, the curve shifts to the right, which means hemoglobin has a reduced affinity for oxygen. So if that curve shifts to the right, hemoglobin doesn't hold on to as much oxygen. How many oxygens are you now delivering to your tissue? 2, 3. Okay? So, what would increase CO2 in muscle? I'm just going to go back to the exercise example. Your muscle's working. As a byproduct, you're producing more CO2. All right? We're going to talk about this in the next couple slides, but an increase in CO2 will always lead to an increase in hydrogen. And if you have an increase in hydrogen, what's that making your body? More acidic, which does what to your pH? It decreases pH. So, using that logic, if I have an increase in carbon dioxide that's shifting my curve to the right, I'm probably also going to have a decrease in pH that's shifting my curve to the right, and an increase in temperature that's shifting my curve to the right. Does that make sense? Okay. All of this would happen in working exercise conditions. Okay. So increase in carbon dioxide, decrease in pH because of an increase in hydrogen ions, and increasing temperature shifts the curve to the right and allows more oxygen to come off of the hemoglobin. Okay? If you go the other way, decrease CO2, then you don't give off as much hemoglobin. I'm sorry, hemoglobin does not give off as much oxygen. Higher pH, don't give off as much oxygen. Lower temperature, you don't give off as much oxygen. All right, this is another video that talks about everything we just talked about. So if you want another explanation of how things go, this is a good video to watch too. Same guy that did the previous video. All right, oxygen is not the only thing we have to consider though. We have to consider carbon dioxide. All right, does anybody know why it's important to get rid of carbon dioxide? I kind of already told you, but I want to see.
Yeah, it makes you go into acidosis if you can't get rid of it.
Right. It makes you go into acidosis. In fact, it is more important to get rid of carbon dioxide than it is to take in oxygen. All right? Because the changes from carbon dioxide to your body are going to happen much quicker than a decrease in oxygen would. All right? So carbon dioxide transport becomes really important if you think about that. All right, carbon dioxide is transported three ways. One is it dissolves in the plasma, like oxygen. Okay, very little of it happens, but it does dissolve in the plasma. Some of it binds to hemoglobin. Remember, hemoglobin is this polypeptide chain plus this porphyrin molecule, and then oxygen's attached. This polypeptide chain is where CO2 would bind to. So we call it carbaminohemoglobin because it binds to an amino acid in hemoglobin. But most of it's transported as bicarbonate ions. This equation right here, if you don't have it already in your memory, commit it to your memory because you're going to hear it here. You're going to hear it again in this course when you get to acid-base balance. You're going to hear it again when we talk about acid-base balance in your Clin Med class. So this is an important concept to understand. All right. How do we convert carbon dioxide to bicarbonate? We have an enzyme, bless you, called Ca, carbonic anhydrase, that takes carbon dioxide and water, converts it to carbonic acid, which then dissociates into hydrogen ions, which is why things get acidic, and bicarbonate. So that's where your bicarbonate comes from. Okay, then what happens, back. Once you have this bicarbonate formed, and again, this is occurring in the red blood cells, this conversion, so you have carbonic anhydrase in the red blood cells. The bicarbonate's going to diffuse out. Okay, then it's going to travel in the blood. And this is part of what gives us our buffer system against acids. All right? What's the charge on bicarbonate? Negative. Okay, -1, but negative is all I was looking for. It's negative. So if I have a bunch of negative ions leaving my red blood cell, my red blood cell is not going to be happy. Okay? I need negative ions to replace those. So what happens is chloride from the blood, which has a -1 charge too, also, will enter the blood to replace that bicarbonate, and that's called the chloride shift. Okay, so anytime in the tissues that you have bicarbonate being formed in the red blood cells, you're going to have this chloride shift occurring as well. All right? So, when you get to the alveoli, let's say that the pressure of carbon dioxide in our blood, the dissolved carbon dioxide, is 45. And in the alveoli, it's 40. Okay? Carbon dioxide is going to go directly from the blood into the alveoli, right? That's how diffusion works. All right? The red blood cells are then going to want to replace that carbon dioxide. So now you're going to have this backwards transition, right? The bicarbonate that's in your red blood cells is now going to be converted back to CO2 to replace the CO2 that's just got lost to the alveoli so that you can keep that pressure at 45. Does that make sense? So the dissolved carbon dioxide is going to leave. Carbon dioxide concentration is going to go down. The red blood cell is going to kick carbon dioxide back out by converting bicarbonate back to carbon dioxide and bring it back up to 45. So you maintain that gradient. Okay? Okay. All right. That's all for gas exchange. Now, how do we control breathing? Does everybody in here have a favorite organ? Yeah? No? They're all pretty cool. My favorite's the brain. Because it controls everything, right? I don't have a heart, so I can't say it's the heart. Ask my wife. I'm not mean to my wife. I just don't really have much feeling. Control of breathing occurs in the respiratory center, and the respiratory center is located in the pons and the medulla oblongata of the brainstem. Okay? In the pons, we have this area called the pneumotaxic area. Pneumotaxic. All right? And its job is to limit inspiration, which means it prevents us from taking too big of a breath and overinflating our lungs. It's involved in increasing our respiratory rate when given the stimulus to do so, and helps modulate the respiratory system according to conditions that it's currently in. All right? That pneumotaxic area can send signals down to the ventral. And what does ventral mean? What's another word for ventral? Anterior. And the dorsal respiratory group, which would be posterior. Okay? So a group of neurons in the anterior portion of the medulla and a group of neurons in the posterior portion of the medulla oblongata. What the ventral respiratory group does, this one right here, it's involved in active respiration, which means I'm consciously controlling my breathing. Okay? So during quiet respiration, which is what you guys are doing just sitting here, that ventral group is inactive. It's not doing anything because it doesn't need to. So most of our breathing activity comes from the firing of this dorsal respiratory group. Okay? And where's that? Never mind. All right. So, in order to control how we breathe, those areas need sensory input from our respiratory system to work. So, there are receptors in the body that control what those centers that we just talked about do. Some are called stretch receptors, and notice that those are in the smooth muscle of large and small airways. So you're going to find them in the bronchioles. All right? What they do is they minimize the work of breathing by inhibiting large tidal volumes. Remember we showed the pneumotaxic area inhibits one of those groups? It's going to prevent overinflation. So that's what the Herring-Brauer reflex is it prevents overinflation of the lungs. Because what would happen if we overinflate the lungs? And then collapse. All right, we also have irritant receptors, nasal mucosa, upper airways, possibly alveoli. What's one important irritating receptor that we talked about? The carina, in the carina. Good. That results in coughing and sneezing to clear our airways. We also have J receptors in the capillary walls that will result in rapid shallow breathing. The important ones that I really want you to know, though, are chemoreceptors and baroreceptors. All right, chemoreceptors are receptors that are sensitive to chemicals, specifically hydrogen ions and carbon dioxide and oxygen, and allow our respiratory system to respond to them. And we'll talk more about those in detail, so I'm not going to go into that now. Baroreceptors are detect changes in blood pressure, okay? And those changes in blood pressure can stimulate us to take more breaths or less breaths, depending on what the blood pressure is, okay? And then we have receptors in our muscles and tendons that will adjust our respiratory activity to the elevated workloads. So let's say we start running, there are receptors in our legs that will tell our lungs, hey, we're doing something really important here, you might want to pick up the pace. All right, so going into the chemical control of respiration, which is what I really want to talk about. Carbon dioxide is one of those things that activates chemoreceptors centrally. So that means that the receptors for carbon dioxide are in the central nervous system itself. Okay? Back up, though. If they're in the central nervous system, that means they have to be in contact with what to detect changes in carbon dioxide? No. The blood. They have to be in direct contact with the blood to be able to detect that. Hydrogen ions, also centrally controlled, same concept. Changes in oxygen are peripherally controlled. So those receptors will be out in our peripheral nervous system away from the brain and spinal cord. Okay, how do these central chemoreceptors work? Well, they're sensitive to carbon dioxide and blood pH, and we already said that blood pH is a result of changes in hydrogen ions, right? Now, even though an increase in CO2 through this equation will result in an increase in hydrogen ions, there are receptors specifically for CO2 and separate ones for hydrogen ions. Okay, so it will respond to both. They're not mutually exclusive, even though the increase in one results in an increase of the other. Okay, so keep that in mind. All right, if we have an increase in carbon dioxide in our body, like for whatever reason, let's say I'm running, what's my body going to want to do? If I have an increase in carbon dioxide in my body, in my blood, what if we're going to want to get rid of it? And so what's it going to stimulate my respiratory center to do? Send more impulses down to what? Diaphragm, which will result in me doing what? Active breathing. What would that operate through? The ventral respiratory group or the dorsal respiratory group? Ventral. Good. Look how far you've come. Okay. We've already talked about this. Okay. What if I have too little CO2? One, is that a problem? Could that be a problem for our body? Too little CO2? Yes. Okay. Alkalosis. Okay. If I have too little CO2, what do I want to do to my respiratory rate? Slow it down, right? So I'm not getting rid of as much. So your job here is to think about CO2. If I have a lot of CO2 in my body, what's that going to stimulate my respiratory system to do, and why? If I have I forget what I just said, too little CO2, what's going to happen? Okay? And this will come up again because we will talk about acid base. All right? So we already talked about all that. All right. Remember we said that the receptors for CO2 and hydrogen ions are centrally controlled in the central nervous system. So those are located in specifically the medulla oblongata. Okay, so when I say central nervous system, I'm talking about the medulla oblongata. Because in the brainstem, is that where thoughts about things can occur? Like if I want to think about breathing, does that occur in the brainstem? No. That's all unconscious stuff, right? Think about it. If you had to, would you be sitting here in PA class learning shit if you had to think about every breath you took? No. Okay, so that's what we mean by centrally controlled. Peripherally controlled, they are sensitive to oxygen. Those are going to be located in the bifurcations of our carotid arteries, where we have something called the carotid body, and in the aortic bodies. All right? Now it's interesting because the other things that are located there, not only chemoreceptors, anybody know what else? Baroreceptors. So these carotid bodies are housed in specific locations to detect changes in blood pressure and changes in oxygen. Okay. Don't worry about this slide. All right. Changes in hydrogen and carbon dioxide, as we saw in the previous slide, directly stimulate these neurons. As soon as they detect the changes, they will fire action potentials, okay, to change respiratory function. The peripheral chemoreceptors, a little slower to act. All right. If they detect a change in oxygen concentration, that'll stimulate a receptor that will close potassium channels. Okay. Go back to asynchronous weeks 1 and 2. You're like, no, please. If I want an action potential to come down here, how am I depolarizing this membrane? Sodium is going in. So what's happening to the charge inside of this neuron? It's getting more positive, okay, because of sodium going in, right? If potassium were to leave, what would happen? It gets less positive or more negative, okay? So the reason I mention that is when oxygen triggers this receptor, it closes potassium channels, which are, before this time, constantly leaking out, allowing potassium to stay in. If potassium stays in, what's going to happen to this cell? It's going to get more positive, okay? So it's going to go towards depolarization. That's going to open calcium channels, which will bind to vesicles and release neurotransmitter to then send a signal to the central nervous system. All right? So is that a direct stimulation? No, that's indirect. So what's going to happen faster? Direct stimulation or indirect stimulation? Direct. So what's going to operate quicker? Changes in carbon dioxide or changes in oxygen? Changes in carbon dioxide, because they directly stimulate the central nervous system, whereas these indirectly stimulate peripheral nervous system, which then has to go to the central nervous system and then do something. Okay? So indirect pathway happens much slower. All right, we're not going to worry about this slide either. See what you guys gain by having short time. All this slide is showing you is that if this is your normal condition right here, this is your arterial oxygen at 100. Remember, it was like 104 in those previous slides. About 100, you're taking about 1 unit of breath. If I decrease that, the more I decrease that, the more breaths I take, regardless of carbon dioxide. Okay, that's what this is showing you. So low PO2, because carbon dioxide is so important in breathing, this is the proof that oxygen can also stimulate breathing changes in breathing rates. Okay, so I'm not just blowing smoke up your ass. All right, respiratory volumes. I'm not going to hold you accountable for all these respiratory volumes except for tidal volume, which is 5,000 liters. Kidding. I will give you guys shit from time to time. The process of acquiring respiratory volumes, has anybody ever done this for themselves? Some. Has anybody ever done it for other people? Yeah? Okay. So basically measuring air volumes. We talked about one respiratory cycle, which is where your tidal volume comes from. And then we always know that there's air in our lungs. We refer to that functionally as our anatomic dead space, but as far as spirometry goes, we call that residual volume. All right, we always have a small amount of residual volume in our lungs at all times for the purpose of, like you said, keeping things inflated. Okay, and this is just a graph showing you all of those volumes. This is tidal volume. When I took a deep breath, that was our inspiratory reserve volume. When I coughed myself to death, that was expiratory reserve volume. And then this always remains in the lungs as residual. All right. I'm not going to go through these definitions because you guys can read and I don't want to insult you. But two things I want to mention. Anything that begins with the prefix EU means normal. So eupnea would mean normal breathing. Okay? Now, what does tachy usually mean? Fast. All right? This is true for the respiratory system and cardiovascular system. If I'm breathing really fast, what kind of volume am I breathing? A high volume or low volume for each breath? Low volume. So the faster I breathe, the faster my heart beats, the lower the volume that's being processed. If I breathe really slow, higher volume. Okay, so tachypnea is rapid because you don't give your lungs enough time to inflate. Okay, so the faster you breathe, the lower the volume. The slower you breathe, the higher the volume. So bradypnea would mean slow breathing. So what's hypopnea mean? Reduced breathing. Okay, you may have a similar volume, but you're not breathing as much. Okay, and then hyperpnea is not necessarily rapid breathing, but it's a higher volume for that breathing. All right, the last concept we're going to talk about is compliance. And this is where my demonstration would have really come in handy. Compliance of the lungs is how well the lungs can expand despite forces that are preventing it from expanding. Okay, so that's determined by how elastic the tissue is and the surface tension. Okay, and this is what we call a compliance curve. I think one of the things I took out of your IOs is draw a compliance curve. You don't have to do that. It's fucking stupid. All right, you guys are going to be PAs, not physiologists. All right, So, I know I'm keeping you guys a little over time. I'm sorry. You guys, do you begin at 3:30? Okay. All right. Here's what a normal lung looks like. A normal lung will inflate and deflate. Inflate and deflate. And we talked about the forces that are involved with that. So let's say it has a compliance value of 1. Okay? If we have more compliant lungs, that means their ability to expand is greater. Okay? What example can we use to mimic what our lungs are like? What could I bring in the classroom that would act like a lung? As far as a balloon, okay? Because it's elastic and it has that recoil back, right? Okay. It's compliant because it's able to expand and retract. If I become more compliant, I expand, but it's harder for me to bring that back. All right? The forces working against and working against deflation are not there, or working with deflation are not there. All right? So if I'm do what? Yeah, it's harder to deflate. So the example I want to give, and like that, if that's an esoteric concept, if a balloon is like a lung that has a compliance of 1, let's say I have a compliance of 2, that would be like blowing up a paper bag. It's really easy to blow up, but you can't deflate it. Okay? And emphysema is an example of a disease where you would see that occur. Okay? So more compliant, very easy to blow up, hard to deflate. If you're less compliant, that means it's hard to blow up. All right, so that would be like trying to blow up a light bulb. All right, bad example. All right, how about a dog toy? How about trying to blow up one of those dog toys? Like, you know, they're tough, they're made out of rubber, you can't blow it up. Okay? Really, that's less compliant lung because you can't blow it up, and it's going to work towards deflation. Okay, so more compliant, less compliant. Easier to blow up versus harder to blow up is what you should get out of that. Emphysema, anybody know what causes that? You guys will get all this in smoking. Specifically, what causes it in the lung tissue? I can't hear you. No? No, but that's a good guess. No. It destroys elastic tissue. Emphysema destroys elastic tissue. So if you destroy elastic tissue, easy to blow up, but I don't have elastic tissue to bring it back down. Fibrosis. How does that occur? Fibrosis means literally scarring. All right? So if I have scar tissue around all my elastic tissue, think of it like putting a fence around a balloon. Can I blow that up? No, but it deflates pretty easily. Okay? So that's kind of a definition of compliance and how it relates to different lung conditions. So, what does that mean? If this is our normal inspiration, expiration, and our normal volume of the lung, if I have an obstructive disease, like any of these, some of which you guys mentioned earlier, I can blow up my lungs, meaning my overall volume is going to be larger, but it's going to stay there, and my overall ability to get rid of air is going to be less. Notice the difference between empty lungs versus empty lungs. Okay? If I have a so we call that obstruction. Emphysema would be an example of an obstructive disease. You get air in, but you can't get it back out. Restrictive diseases would be like fibrosis, where I can't get much air in the lungs, so my entire volume is about 3, whereas normal is 6, right? I can't get air in, which means I can't put much air out, and therefore my ventilation is compromised. Okay, does that make sense? All right, that's the last slide, right?
Yeah.
All right, you're done. A pneumothorax can cause an atelectasis. Yes. So a pneumothorax would be a hole in your chest wall leading to a collapsed lung.
Oh, okay.
So, with restricted lung disease, one of the issues is like a higher concentration of oxygen that goes to scarring because it's more resistant to oxygen.
I can't hear you. Say it again.